So biotechnology, go figure, is using biology to produce a better product, to produce a better world. We are capable of changing our environment. We're one of the few organisms out there, us and beavers. We change the environment to suit us. And we've gotten to the point where we can not only change the environment, but we can change other organisms. So it's the application of technologies that involve the use of living organisms or products, or products from other organisms. That is, we use organisms to make other organisms. Milk, cheese, yogurt, beer, just about everything out there that we ingest is going to be a product of biotechnology. Now, we've gotten more advanced lately, but it's still the same process. We started off with domestication. Domestication is sort of the penultimate form of biotech. You take some living organism out there that had one role, and you change its role. Okay, so what was the whole point of, what did horses used to do before we got involved with them? They ran wild. What does that mean? They were rabid? They lived as they pleased. They ate all sorts of grasses, things like that. We got horses, and what did we do with horses? We ride them. We made them work. Now we don't have to do that work because the horse is doing it. Dogs. We took... dogs are like the ultimate story of human ingenuity. We took our predators and made them our friends. Not only our friends, but they do work. You guys... some of them do. Okay, your pets probably don't. But let's say like a Labrador Retriever. Anybody have one of those? No? Golden? Okay. You know what? Yeah. Those things will grab anything with their mouth and just carry it around, like for fun. We've engineered that into them. They didn't used to do that. They didn't bring them back. Cows. Can you imagine a wild cow?
I know there's no more wild cows anymore.
Right. But they came from wild stock. Right, the same thing, but we tame these. Chickens. Again, imagine a wild chicken. Velociraptor, wild chicken. This is teosinte. This is the baseline corn. This is what corn used to be. And then we got it, and we made it into what we know now. It used to be very, very small kernels, not very sugary, definitely not white, very short grass. No, it was terrible until we got a hold of it and worked on it for 1,000 years. And now it's all over the place. And it's only all over the place because we decided it was going to be. We've domesticated it. Just about every organism out there that you deal with on a regular basis has been domesticated. No. Well,
domesticating foxes.
Ah, well. So we've engaged in biotechnology for years and years and years and years. Now we're in this modern world of biotech where we can specifically genetically engineer microorganisms. We can make it very, very specific what we want to change. What used to take thousands of years of random mutation can now happen in a year or 2 years. It's still the same idea. Whether we're taking corn and it's getting mutated by ultraviolet radiation from the sun, or we're taking corn and mutating it through ultraviolet radiation in the lab, same process. Heck, we could even go faster. Instead of using ultraviolet radiation and having random mutation, we can actually make it site-specific. We can look at it and say, we want to hit that gene right there and see what happens if we mutate it. Same process, it's just happening a lot faster. A genetically modified organism, otherwise called a GMO, is any organism that's received genetic material through recombinant DNA technology. There you go. So anything that's gotten foreign DNA placed in it, and that's most things now. Chicken nuggets, whatever. I'm thinking, like, if you think about plants. Tomatoes. We've engineered genes into tomatoes to make them more resistant to cold. We've engineered, and to a lot of plants, we made them more resistant to cold so that we can continue growing them through the winter. Right. We've made things that used to be bushes into trees. We've made things into we've made all of these leaps forward. Transgenic organisms, plants, and animals are going to receive genetic material from a different species. So there's a difference between genetically modified and transgenic. Genetically modified is the DNA has been altered. Transgenic is you've taken DNA from one organism and put it in another. If you were to take the green fluorescent protein from a jellyfish and inject it into a mouse egg, eventually you end up with a specific glowing mouse. Pardon? It does not attack it. It recognizes them as your own. Right. It has to be introduced into the genome right early on. There's a lot of potential. There is, for every success you have, you're looking at 1,000 failures. Now, this is obviously big business. Recombinant gene technology and transgenics really came in big in the late 1970s, early 1980s. That's when scientists realized they could do it. They can change genes. And in the 1980s, the court said you can now patent transgenic animals. So now you don't just have to let it, once you've created it, it's yours. That set of gene is your property, even if something else has the genes. You could sue somebody if they have a glow-in-the-dark mouse that used either your technology to build that mouse or they used the gene that you patented. And then you would get any product that they would have been able, any money they would have been able to bring in. It is. It's exactly. But they've done that.
So how much money per year are you making on royalties from your?
Nothing, because I use somebody else's technology. I know. Now we can use microorganisms, genetically modified microorganisms, for lots and lots and lots of different things. We make medicine, fermentation, biological control procedures, and bioremediation. As far as medicine goes, we used to extract insulin from pigs. We don't do that anymore. We used to take the pigs, remove some of the blood from the pig, spin it down in a centrifuge, extract the insulin, and then use that to treat people who have diabetes. They stopped doing that about 15 to 20 years ago, and they switched over to having E. coli make it. Because instead of using pig genes, now we're using human genes. That's right, but we've engineered human genes into the E. coli. So it's transgenic. You're producing a ton of insulin now. Right, well, now it's changed slightly. It's very, very efficient. You're producing a ton of it. You're producing human insulin, which means there's going to be less allergic reactions. More people can partake, and it's a lot cheaper. As far as food goes, any microorganism that we use for food production, whether yeast making beer, any bacteria used for fermentation, any when you're canning things, these organisms are, for the most part, genetically modified. If you find a beer, you're going to find that most of them have to be labeled as genetically modified because of the yeast that they use to produce it. They've made them, and they're terrible. Biological controls. We can use microorganisms to control other microorganisms. No, those are, so a parasitic wasp will have stung the caterpillar and injected its eggs. And then the wasp's larvae eat their way out of the caterpillar and then spin their own cocoons on it. They should stick to figs. But you can control these caterpillars, these hornworm caterpillars, through by introducing these wasps, these other organisms. That's biotech. Using bacteria to fix nitrogen. We spray bacteria on fields now because bacteria are nitrogen fixers. Plants can't do it. So if you have a field that needs a lot of nitrogen, like, say, a cornfield, instead of going and putting fertilizer out, you spray it down with bacteria. And those bacteria take nitrogen out of the atmosphere and produce what plants need. Exactly. Again, another example of, right, it's another example of biotech. In bioremediation, you take microorganisms and you could spray them down with the areas that we've screwed up. Right, we have not just mushrooms, but they've taken entire oil slicks. We have bacteria that devour oil. So we spray down an oil slick, and that oil gets degraded. There are beaches that have been utterly destroyed by oil spills. Black beaches. You've seen images of like otters and ducks and things coming up, and they're all covered in muck. We can spray these beaches now with a bacteria that will break down that oil into its constituent carbon components. So it goes away. Bioremediation. We've designed all of these organisms. You guys have probably heard of stem cell research, right? Stem cell research has a lot of potential to it. What a stem cell is a cell that has the potential to divide and divide and divide into any type of different cell. Totepotent ones arise right after fertilization. So back to birds and the bees, you have an egg and a sperm, and they come together and they fuse to form a zygote. After that zygote forms, it will start to cleave or split. So you go from one cell to two cells, 2 to 4, 4 to 8, and so on. And those cells can turn into any cell because they are sort of blank programmed right now. Eventually, they'll differentiate. But before that, they're known as totepotent stem cells. They have the potential to be anything, any type of cell in your body, including brain cells that you don't get to replace. So if you were to, the idea is if you were to take a stem cell that was compatible to your body and inject it near another cell, it will copy that other cell. It will make itself into a type of cell like that.
Is the DNA just like some sort of like a communication that goes on between them?
There is. You have to pass information between the cells. So you're going back and forth between the cells. So if one cell is going to be one thing, the cell next to it is going to, or the totepotent stem cell would adopt that shape. Would you be the same person? It depends. So, right, but, well, the issue is these cells are going to be slightly different than your cells because they're going to not be, unless you've got totepotent stem cells left in your body, which you probably don't, they're going to be somebody else's cells. Then that's different. And that's where you get ethical implications, when you start cloning, where people say, I'm going to create an organism exactly like me with my DNA that I will then harvest for its stem cells.
See, there's ethical ramifications either way.
There are.
Depending on what your perspective is, my dad needs some stem cell treatment for like an inflammatory disease, but he won't do it because he doesn't believe in it.
There you go. So those are totepotent stem cells. Every single cell in a plant is totepotent. Every single plant cell can become any other plant cell. Humans and other animals are different. Typically, our totepotent cells disappear early on in development. They become fixed. That's where you get this idea of the embryonic stem cell. These lead to pluripotent cells, cells that can become almost anything. And this is probably what you hear about most in the news, where people are talking about embryonic stem cells. These cells can become nearly every type of cell in an organism. But the problem is, when you're harvesting them, you do destroy the embryo. So again, there could be ethical issues. That being said, we destroy a lot. I know it sounds really bad to say it, but we destroy a lot of embryos all the time, especially with in vitro fertilization. Well, it's not just that, but say a couple goes in to they want to have a child, so they go in for in vitro fertilization. They're not going to produce just one embryo and say, we're trying this. They're going to produce 12 or 15, because not all of them take. Well, once they have that, once you have a take, what do you do with the rest of the embryos? You freeze them, but then you have a maintenance cost year after year after year.
You can't just like give those to somebody else because it's not their egg.
You can't, right. What?
Basically throw them away.
what ends up happening is they become medical waste for the most part.
And if you have permission, you can turn them into embryonic stem cells.
Correct. Now, less ethically questionable would be multipotent stem cells. You have lots of cells that are termed multipotent. For instance, you have stem cells that produce blood, hangs out in your bone marrow. If you lose it, you don't make blood cells anymore. So what do you have to get? You have to do a bone marrow transplant. Why do you need a bone marrow transplant? So you can keep making blood. Right. You've got stem cells that produce skin. They're constantly producing new skin. So we do have some multipotent cells, and in some cases we transfer them back and forth to each other if we're compatible. And then you've got unipotent stem cells. These are cells that are found in adults, and they can only become one type of cell. In your testes, well, not all of you, but in some of you, you have testes. These are going to produce sperm through your entire life. You've got a stem cell that keeps making more and more and more of this one type of cell. So these can be used for treating a lot of different diseases. Anything that causes deterioration, like Parkinson's disease, spinal trauma, joint damage, bone breaks, liver failure. These are sort of a medical almost cure-all. You can literally make new teeth. Yeah. No, you have to. Well, you have to inject them near a cell that is similar to the one you want to copy. So that's how lizards regrow their tails. Regeneration.
So in order to make a new tooth, one has to inject some stem cells into the old tooth.
Right. But then you could just implant that other one.
It would grow out of the tooth? Or
No, you have to plop it over.
take it and then insert it into the
Right.
nerve cavity.
Okay, so that's how, so medical marvel for the future, and that's something that's really on its way. We're very, very far advanced in that field. Genetically modified plants, another thing. These are on the market. You've probably seen and heard of lots of people debating these. Over time, we have modified and bred plants to create lots and lots and lots of new types of crops. Just about everything that's out there that you eat was not what it looked like originally. Well, there's we only eat female corn? I see what you're saying. Right. You've got different corn is it produces the male part of the plant is in a different place than the female part of the plant.
Oh, the male's at the top and
Right.
the female's all over. Okay, so it's what's it called?
Perfect. That's what it means. It's the perfect plant. So we've created these new types of crops. You guys have seen tomatoes, and you know they didn't look that huge thing before. If you've ever gone walking in like some wild-looking woods, you don't see giant fruits hanging from trees. Ooh, except the pawpaw tree. Have you ever heard of the pawpaw tree? It might be called the horse apple. I've decided I'm getting a pawpaw tree. I'd learned about these. They're native to Virginia. You don't find them often, but they make these large fruits that are really sweet that only last for like 2 days before they rot. So you can't sell them, but I could have them at my house. I think they're green. What? No, not those. Those things you just throw at each other. That's what those are for. Crab apples. We can modify plants not just using the old way of using ultraviolet radiation from the sun, but through lots of different ways. Agrobacteria transformation, biolistics, microinjection, and protoplasts. Pretty much, you take those totipotent stem cells and you trick them into doing what you want. For agrobacterium, agrobacterium is a natural bacterial genetic engineer. It is way more efficient than we are. We've been trying to perfect genetic modification. These guys did it a long time ago. What they do in their natural state is they induce tumor formation in plants. They effectively cause plants to have cancer. Because these tumors are going to provide a house for the bacteria. So what we did was we looked at that and said, well, in order for them to do this, they needed to trick the plant into expressing bacterial genes. Which means that if we can modify the bacteria If we can change the genes in the bacteria from build a house to glow green and infect the plant, then we can make the plant glow green. So what you do is you change the bacteria. If you've changed the bacteria, you genetically modified it, you've added a new gene in, you then get the bacteria to infect the plant. The bacteria inserts your gene into the plant. Now you have a plant cell that might glow green. You make that plant cell grow. You give it some, we haven't talked about like auxins and cytokinins. That's next semester for you guys. You give it hormones. You guys know what hormones are, right? You've experienced them. Hormones will tell the plant, grow roots, grow shoots, until you get a plantlet that eventually becomes the transgenic plant. Every single cell in this plant, produced by that plant, produced by that one cell, is going to express your gene. Fully, yeah.
Do you think they'll ever change pineapples to pink pineapples?
To pink pineapples? Why would they do that? Do you want a pink pineapple? Is there a market for a pink pineapple?
I would like to, like a pink lemonade.
So pink pineapple, you could just add a dye.
But there aren't no pink ones.
Ooh. You could engage in, honestly, I don't know about making a pink pineapple. If there was a market, somebody would make it.
I know Texas A&M University is really good about making multicolored vegetables and fruits. They probably have already made
There you go.
maroon pineapple. I know they make maroon carrots, and they made some flowers that only come in orange and white. They made those purple also.
Okay.
So they're all about that.
Another way to modify a cell is through what's called biolistics. And it sounds sort of like ballistics, right? Because that's what it is. You load up, you coat little balls with DNA, the DNA you're interested in sticking into the organism. You load them into what we call a gene gun. Not kidding, that's what it's called. And then you fire it at your cells. It does. It's called a shotgun approach because you are blowing holes in this genome and hoping that some of your DNA sticks. It doesn't work often, but if you're dealing with 100,000, a million cells, and you hit them all, it works. You get one or two that work, and then you've got a transgenic cell, which you can then cause using hormones to grow shoots, grow roots, and then you have a whole plant. This also works on other animals and such, but again, the success rate is so low, it's really not worth doing. It's expensive. Microinjection is more precise. It sounds just like what it is. You take a needle that's got your you got your DNA in solution, you get it through the cell membrane, and you inject it directly into the nucleus. Sometimes it works, sometimes it doesn't. I'll reject you. And you could use these naked cells. This is where the cell wall's removed, which makes it really, really easy to just trick something in through the plasma membrane. Protoplast. This one. You could have lots of uses of transgenic plants. This is a photoshopped image. We don't have blue strawberries. We could, and truthfully, it wouldn't matter much. But the thing is, in a lot of plants, you can't get blue to express. I've tried. Blueberries, right? You can. What? That's a good question. We can use transgenic plants to protect the plant, to stop organisms from eating it. Plant quality, we can make it more sugary, more delicious, or create new products like pink pineapples. Yes?
So what does transgenic mean?
Transgenic means you are taking a gene from one organism and putting it in another. You put blueberry DNA into a strawberry, though that's not what happened. Question? Okay. Blueberries are good. So you can protect plants. Take a look at this field. This is a transgenic experimental plot. And you'll notice you could see the transgenic plants, transgenics, non. The control plants, the non-transgenic plants, are not doing as well. In this case, they were testing drought. So they created drought-resistant plants. Cam plants, you could think of it that way. Plant quality. You increase their nutrients. You make them better. I used to do a thing called strawberry nutrigenomics. Literally, we were trying to figure out not how to put stuff into the strawberry, but what genes in the strawberry controlled what nutrients, so that they could be modified later to increase or decrease that nutrient load. Yes. You could come up with new products. That is also Photoshop. No, that might not be Photoshopped. They just cut it out and put it in, yeah. I should really do that, like at the beginning of a 101 class, now that I think about it.
There's this stuff that's like, it's called meat glue.
They would. They would totally. Okay, next semester. For those of you who will be in my 102, I'll report on this and let you know how it worked.
There's this stuff called meat glue. If you do it right, all it has to be is kind of wet and it'll glue stuff together, so you can make it look like it's a full orange.
Oh, I see. Right. I'll try. Meat glue. Now that I know that exists, not going to forget it. We actually are using plants now to create plastics as well. We've modified them in such a way that they can produce proteins that can mimic plastic properties. They break down. And so you guys may have heard of gene therapy before, right? What gene therapy is where you introduce clone genes into living organisms. You're introducing foreign genes in. Some of you guys, and I'm not naming names because I don't know and I don't care, have genetic issues and that you might want to have fixed. This therapy may be able to help you in the future. Typically what they're going to do is create a liposome made up of a bunch of phospholipids. And inside of that, you're going to have your recombinant DNA. So this liposome gets injected into your body. It gets pulled into the cell through endocytosis. And when it's pulled in through endocytosis, the liposome breaks down, releasing the drug, whatever that drug may be or whatever that DNA may be. It's very, very specifically targeted. So if you needed to hit brain cells or spine cells, you can make it by adding the right recognition protein on the outside of the liposome. So it delivers drugs only to the cells that you need it. They're also looking at this as a way to combat cancer cells, because cancer cells produce a certain recognition protein. So if this binds only to cancer cells, it can deliver a lethal dose. We could also use viruses for this. People have made inhaler sprays where you've engineered a virus. That virus no longer has viral DNA in it. It has your modified DNA. They did this with the flu, yeah. Yeah. That is not what inhalers are, no. The flu mist is this?
Question.
Yes.
Don't viruses have just RNA?
Some viruses have RNA, some are DNA viruses. And so we can use this recombinant DNA to move fragments of DNA around into new arrangements. That's how we got the glowfish, the ones you can just go to the store and buy here in Virginia. There are a lot of states, by the way, where it's illegal to buy glowfish. Yeah. Because they're transgenic.
I bet they do. I bet they die the next day.
That's just because you're terrible at raising fish. We have two types of techniques we can use, in vivo and in vitro. And there's a difference. And it's a really easy question to say, what's the difference between in vivo technology and in vitro technology?
Well, there's a petri dish on vitro.
Right. In vivo is you're injecting it into a living organism. In vitro is where you alter DNA outside of the organism. So in vivo is directly getting the DNA into the organism, and in vitro is you deal with it outside and then you inject it in.
So for the people that are getting in vitro insemination, right?
They create the organism outside the body.
And then they put it in
Correct.
in vivo.
Right. Well, it would be in vivo if the procedure was done inside the body. But it wasn't created inside of her. It was created outside.
So in vitro, you just planting a fully fertilized zygote.
Right. Now, back in the 1970s, this is where all this biotech came about. You're at Stanford University, and they took these two isolated DNA fragments and bound them together. So you took DNA and stuck them together, and nobody knew that could happen until the 70s. It was huge, because once we figured out how to connect two pieces of DNA, and it still transcribes, it still replicates, now we can start changing the structure, the fundamental structure of our instructions. So after they built this DNA, they stuck it into a bacteria, and that bacteria started making whatever that gene product was. So they created a new polypeptide. This is the real structural foundation of transgenics. And I tell you that because this helps us understand, this ultimately for all of biology, it was a huge leap forward. We don't have to just observe what's going on. Now we can directly experiment with genes. We can turn things off, we can turn things on, we can figure out functions. And that's what led us to be able to interpret what all of our instruction manual says. That led to the field of molecular biology, where we're routinely using cloning and genetic transfer technique in order to figure out how the world works. Gene cloning is where you're going to make lots, and go figure, right? You're going to clone something. You're making lots of identical copies. Now, with gene cloning, you can copy genes left and right.
Left and right, just
Either direction. Actually, it's true because you could copy either side of the DNA if you knew what you were doing.
the 3' or the 5'.
Right. So it helped us really start understanding the genome being the entire set of instructions to build an organism. Now, some genomes are complex, like yours, and some are very simple, like a virus. I want you to understand the difference between the three genomes. First off, we got viral genomes. Like you were saying, viruses contain their entire genome inside this teeny, teeny, tiny head. They're really small. They're a fraction of the size of a cell, like 1/1000th the size of a cell. Yet they have to keep their DNA in there. That means that these are very, very, very small genomes, about 100,000 base pairs or so. Very small. If it was bigger, it wouldn't fit inside of a capsid. And we don't have time. I wish I could tell you. So you guys know about viruses, right? What are the big viruses you know? Humans. The flu. The flu is very variable. You'll notice that every year there's a new strain. Every year the flu adapts. And the reason it's able to is not just mutation, but it's due to what's called alternative splicing. You have one big, long gene sequence, but it may only express two things at once, or three things, or 1 in 3, or 1 in 7, or
Sometimes it's airborne, sometimes it's hypercontagious.
right. These different combinations trick our immune system because we're ready for one particular type of virus. You are set for last year's flu virus, but it's gone now. There's a whole new virus that's replaced it. So your body has to readapt. So next time you get sick, your body begins to produce antibodies against that particular virus, and then the next year it comes along and it's changed. Moving up a little bit more complex are the bacterial chromosomes. Bacterial chromosomes are held within the nucleoid region, because remember, there's no nucleus. And they are inevitably round. Bacterial chromosomes are circles, and they're about a few million base pairs in length. And then you have your eukaryotic chromosomes. It's not just one chromosome, now it's a series of chromosomes in the cell. These are usually huge. They're a gigantic size. Instead of a million base pairs, now we're looking at 10 million, up to 1 trillion base pairs. bacterial genome versus eukaryotic genome. Somewhere. In order to sequence the genome, we've got to clone bits and pieces of it, and then run these cloned pieces on what's known as a DNA sequencer. We used to have to do it on a bunch of gels and do it all manually by hand, but now we get computers to do it, so there's not really much of a point in talking about how it happens. It is. It takes months. Each little tiny piece. The human genome is sequenced now. We know basically what our manual says, how to build a human. Not how to build a human. We know technically the genetic sequence, but we don't know what every piece does and how they relate to all the other pieces.
We only have like 15%
Of it known. We know the whole sequence, but we don't know what it does.
We know 15%
Right.
and how it expresses.
It took 20 sequencing centers about 13, 14 years to get this done. It was expected to take like 40 years.
You think about the processing power that would have taken in 1990 compared to like now? You literally do that on your smartphone.
Well, you can't do it on the smartphone. It still does require stronger computer technology, but we have been sequencing things. What took 13 or 14 years then will now take 6 months.
A couple hours if you had 20 countries.
That's true. If you have that many. But the technology has increased so quickly, beyond any doubt. We are leaping ahead as far as technology goes. It's giving us an enormous edge. I just recently, I don't know about you guys, in my house I have this one drawer where all my random electronic cords go. Yeah. Well, and it was filled with these cords, and I was getting ready to move, so I was like, I got to get rid of these things. And I had a bunch of old hard drives. And I was like, I don't know, maybe I'll still need these hard drives someday. And then I looked at them, and they were 128 megabytes. That was a lot of data for in the day. And then I was like, there's no way I'm ever going to look at this drive again.
I just bought a 16 gigabyte drive for $7.
There you go. So back in the 1980s, we could clone about 1,000 base pairs per day. In the 2000s, we can clone about 1,000 base pairs per second. So huge difference. Yeah, for a lot less money. Look at that. 2003, it would cost $100 million to sequence the entire genome. Today, it would cost $1,000. Literally, it doesn't take much anymore. And because of that, because it's so quick and it's so cheap, you can have people doing this literally in their backyard labs. No, there's, I read a story about this one idiot who swore he was a geneticist. He had a lab in his yard that he made for like $5,000, and he had gotten a hold of some virus material, and I know, this is how it all ends, right? He'd gotten a hold of some virus material and some, like a hormone sequence, because he decided he was going to make himself stronger. So he injected himself with his homemade concoction of genetic mutation. Yeah, it was a bad idea. The whole goal of this project, the Human Genome Project, was to identify the function of every structural gene and the impact of all the regulatory genes, the activators and the repressors. Figure out how they all work together. We are not there yet. We're getting closer, but we're not there. Because after we start looking at the Human Genome Project, we have to look at the Human Proteome Project. This is identifying all of the proteins expressed in a cell at any given point. Because for every gene you've got, you could have many, 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 many more proteins created. Hundreds of years. Yeah, we say that now, but 40 years from now, the processing power may be so much higher.
Have you ever read The Last Question by Isaac Asimov?
No. Okay. So if our genome is down on this end, the complexity continues to increase almost exponentially as you start to look at the proteome. So it gets a lot more complex. But that's about enough for introductory biology. Now that I've completely scared you away, from any sort of future endeavors in bio. The whole point of this is to make people's lives better. You got to remember that as you go forward, if you go into bio, whatever field you go into, do it to be good. Don't be evil. I think that was one of the first lessons we learned at the beginning of the class, right? Don't be evil. Be a good person.
Don't be a mad scientist.
Don't be a mad scientist. Try to make the world better. So, do you guys have any questions? Here are the content review questions. I hope you enjoy them.